Good, nice. So let's begin. At the beginning, thank you really for giving me the opportunity, guys, to share my paper in quantum biology. So I am making a little introduction of myself, because at some point stuff will become super weird. So you can understand that I am a real scientist and not a totally crazy person. So just for you to understand, I have like the normal, typical scientific career. Have a degree in biology, 2009. Then I have a PhD in biotechnology in Spain. Then I have a postdoc. I did already 3 postdocs, 1 in Spain, 2 in the US, between Cedars-Sinai Medical Hospital and UCSD in San Diego. Then I create a company, but I have like 15 years doing science, not in quantum biology, but in metabolism, in cardiology. So I have a real record, about more than 20 papers, 900 citations. So why I am doing this? Because of that, because stuff will become weird in some point. So you can believe that I can be a functional scientist outside quantum biology. But and this is my current group. I am based here in Argentina with my company, which is called SheCorp, working as well for the cardiometabolic lab, which is in Denmark, in the Aalborg University Hospital. That was the last paper that we published about some proteins and working in metabolism. But during these last 15 years, I teach myself about quantum physics, quantum mechanics, mathematics. I try to do that. And that ends in this paper, which is called DNA as a Quantum System in Evolution. So to begin here, this paper tries to answer this question: how does life or living things measure time, right? That is the main question that I am asking here. So we already know about some biological clocks that are useful as readouts, right? But that do not mean physical input. What I am trying to say, telomeres, for example, are the tips of the chromosomes. And you know that with aging, telomeres are getting shorter each time. So people believe that that is a mark of aging, and which in fact it is. Then you have epigenetic clocks, circadian rhythm. I mean, we already understand something about aging in cells, but the deeper question is, what physical variable is being converted into these downstream age marks? Because that is what it is, aging marks, not a way to measure measure time, really. So the question is, for me at least, right, is when an organism begins to age. And to me, the answer, again, is from the moment it begins to exist. And that moment is always, from my biological perspective, linked to DNA as a moment zero. If you are a bacteria, you need to replicate. If you are a mammal, you need to go through the union of two gametes. But DNA is always in this moment zero. So for me, it is logical that DNA needs to do something in this process of how to measure time. And this is really, really important, and we need to pay a lot of attention here. Across different species, including even bacteria, some mutations, forget about somatic mutation rates, scale with lifespan. What that mean? That means that, for example, any of you guys that are listening this talk have more mutations in non-coding regions in your DNA than 1 year ago. This is not me saying that. This is hardcore science. Nature and Science papers already show that when you age, you accumulate mutations in non-coding regions. Your DNA have 2 regions, coding and non-coding. If you introduce a mutation in a coding region, that is called cancer or a tumor, right? But if you introduce that in a non-coding mutation, in a non-coding part of the DNA, you have a really, really huge chances of nothing happens. So to me, and that is a mark of aging, really. So keep that concept in mind. So sorry about this, but we need to introduce some physical and critical concepts that at the beginning will be like separated, but step by step we will start to put all the pieces of the puzzle together. I need you to understand what Doppler effect is. It's a really easy physical phenomenon to understand, and it is related with how a wavelength propagates, and it's related that if the source of a frequency, a frequency, is in relative motion with the, in this case, a person which is receiving this frequency, the frequency change respect to the movement of the object which is emitting that frequency. The typical case, to make an example, is the ambulance. If you are in the street stop, the ambulance is coming, you listen one noise, and when pace in front of you and go away, the noise changed. That is because Doppler effect, and we need to keep this concept in mind because it's key for understand the paper. So then, this is another interesting concept, which is informational entropy, and how that informational entropy is related with the Doppler effect. Let's say that time is not a magic substance, and it is the direction in which physical systems accumulate histories. I mean, we already know that the universe is growing and expanding in an accelerated way. That means that the amount of informational entropy increase each second, each millisecond, right? So again, this is not me. This is physics talking. Really more smarter people than me. A way to understand how times is going forward is because we are able to measure this informational entropy increase because second law of thermodynamics, right? Because universe is expanding. So if we mix the expansion with the Doppler shift, what we saw is that there are some radiation from the beginning of the universe, which is called cosmic microwave background. This kind of radiation that is approximating to Earth, and in this case to Earth DNA, as the universe is expanding, expanding, that signals are moving away from us. So it is coming with a signature of time encoded in this Doppler effect. Because of that, when we saw the stars, all the light is moving to the red because the frequency is going down because the galaxies and this microwave background is moving out from us. I hope that I was clear with that concept because it's key as well. That is the same. Now, another piece of the puzzle. DNA can act as a fractal antenna. Again, this is not me. There are really nice papers. There are some people that like it, some people that no, but to me there are nice, strong papers claiming that DNA had fractal antenna properties, right? In which range from 1 to up to 1300 or 800 gigahertz, which is kind of the same signal of the cosmic microwave background, right? So if DNA can act as a fractal antenna, maybe it is able to receive this frequency to measure time. That is the main hypothesis. I need to jump because I am taking really a lot of time to explain this, and I will run out time. So let's go with the main hypothesis already. I hope that the evidence was enough, but the main hypothesis, right, of the paper is that maybe DNA with their fractal antenna properties use this cosmic time. Sorry, I need to show it. Okay. Use these cosmic signals to measure time. In which way? By measuring this Doppler shift. Okay, so we have this CMB, this microwave input, which is already Doppler shifted because the expansion of the universe, and in that physical phenomena is encoded the time, right? Then DNA as a fractal antenna will be able to measure this. And here is important, as there are really nice papers talking about the quantum properties of DNA, I am proposing here that if DNA acts as a quantum system using these fractal antenna properties, could act as well, as a kind of a quantum computer, a quantum system to process these signals in order to measure time. As we already know that a way to measure time is introduce mutations in the signal, in the DNA when DNA is replicating, my hypothesis is that DNA will be able to measure this Doppler effect to acting as a quantum system, introduce mutations in the DNA, right? By definition, a computer needs three stuff: input, processing, and output. So to be clear here, the input is an electromagnetic signal. It can be CMB, it can be, now I already working in other stuff because this is too speculative and I figured out some really nice stuff that I will tell you after. Then you need to simulate this input as a quantum system, and the output could be the altered properties of that quantum tunneling occurs. Why quantum tunneling? Because quantum tunneling is a phenomenon that is really directly related with the probabilities of the polymerase when replicating to introduce mutations in the signal. I sorry if it's too much, but that is all the puzzle, right? So this next slide is about this one, right? The replication outcome. So there is a quantum phenomena which is called proton tunneling, which is already related with another quantum phenomena which is called tautomeres. I don't want to become too technical because the talk never ends, right? But let's say that DNA is not a dead book. It vibrates, resonate, and exchange energy. So it's true that the insane sounding hypothesis is built from non-insane pieces. What I'm trying to say here is that proton tunneling, yes, is in the background of how, again, this is not my idea or hypothesis. This is really nice science and papers that already demonstrate, but in silico and in theory, that this phenomena, which is called proton tunneling, is related with the chances and the probabilities of introduce mutations in a sequence, in a DNA sequences, when polymerase is replicated. So I will stop here. I don't want, again, to become technical, and I don't want to start to explain what proton tunnel is. I mean, you have some energy barrier. If you are a proton, for example, which is in this case, some protons which are sharing between the bases of the DNA, adenine, thymine, cytosine, and guanine in the double helix. So if you pass some barrier, you are able to go to the other stage, and when the polymerase is replicating, you have micro timing. The correct way, the word here and the concept is micro timing. So you are the polymerase, you are replicating, so you have, let's say, you need to copy the DNA that you have in front of you. So if you have a thymine, the correct base to introduce is adenine. So imagine that if you need to introduce in the sequence adenine, and this happens a lot of time, you introduce another thymine. So the proofread mechanism of the polymerase, now I am explaining the whole thing. the proofreading of the polymerase have a micro timing, which are femtoseconds, nanoseconds, is really, really small. We use these proton tunneling energies to, let's say, to measure, because this become weird, but you have a micro timing where the polymerase decide if use the proofreading mechanism or not. So if you go through this barrier, the polymerase, let's say, became a mistake and introduce a mutation. If not, you have fidelity, right? So we have this quantum phenomena, which is called proton tunneling, which is already linked to mutation. So what I will sham, sorry. So this is what the paper already measured, right? The first stage, I mean, now I need to start to try and to test all these ideas, in silico and doing simulation. So the first, what I did, really the second, is try to measure von Neumann entropy. What is von Neumann entropy? Is how mixed the DNA quantum state is, really. Again, this is hard, but the takeaway message here is that, okay, I take the the tuberculosis genome, which is, for the size and the computational power that I have, is what I can do. So I take that genome. Are you listening me until here? Are we okay?
Yes, so far so good.
Yeah. We are okay.
Yeah, keep going.
Okay. So I take this genome and I make the decision of map the nucleotides of that genome to different quantum, to qubits, really, to qubits, and simulate this von Neumann entropy with a code in a computer. And what was the result? That von Neumann entropy shows that coding and non-coding, because the first step was to divide the sequence in coding and non-coding, and then assign this von Neumann entropy stuff, right? So what I saw is that von Neumann entropy showed that coding and non-coding DNA remain distant even after quantum invariant mapping, because this is a limitation. Don't care about that. So the takeaway message here is that if you understand, I mean, this is a good result. If you understand the DNA as a quantum system, which is what I am trying to say by applying this von Neumann entropy model, you already can see difference between coding and non-coding regions, right? So it seems that if we understand that DNA is a quantum system, at least at the beginning, coding and non-coding regions behave different in a quantum way. That is what this results say, right? Then the next step was to simulate the quantum dynamics. I mean, how this DNA of Mycobacterium tuberculosis evolves in time if we understand that it is acting as a quantum system, right? Again, I split the genome encoding and non-coding regions, and without adding any external signal, which is the CMB, cosmic background, blah, 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 the paper and the result shows that real coding and non-coding DNA already behave different in a quantum-like dynamics. This, guys, is a lot. This is really a lot. I mean, maybe do not sound like so crazy, but to me, it's amazing because, again, here you have the evidence with all the limitations that we can talk after, but you have a real evidence, a super strong p-value, that if we model it in a computer, we make a computer simulation with these parameters, right? About trying to understand how DNA evolves in time, we already saw really interesting properties. Which are those properties? Coding and non-coding regions behaves different. And and this is all again controls, right? If I use fake sequences or control sequences, the results are different. So this is really nice. Then the other stage was try to see what happened with this evolution of the quantum system, that now the DNA, is our quantum system. What happened with that if we introduce an external signal that are able to modify this quantum behavior? And again, you code all of that, you make simulation. I mean, these simulations take 3 personal computers or 4 during 6 months. I mean, it's long simulations just for 200 base pairs DNA. I mean, at the beginning, von Neumann entropy was with the whole genome, but for this, you really need a lot of computational power that I don't have, but still giving some really nice results. So if we increase that, I think that the result will be even stronger. But okay, sorry, going back to here, the takeaway message here is that when the input signal, because you can model all of this, in the code that you write and simulate in silico, when the input carry the Doppler-like time code, real DNA organization produces a stronger spectral response than controls. What that means? That if I use real DNA sequences and I perturb that real DNA sequences with this 34 gigahertz cosmic microwave background, the real signals are more responsive than the controls. Again, reinforcement the concept that DNA can act as a quantum system. So the final, and I am finishing here, is how this input signal encoding time through the Doppler effect, because you can simulate again the Doppler effect in the code and in the simulation, how that was altering the probabilities that quantum tunneling occurs when DNA replicates. So I aside that, let me try to introduce, because maybe I don't want to lose you guys, but the concept of proton tunneling was that when polymerase was replicating, if you go through a certain energetic barrier, the polymerase can become mistakes and introduce mutation, and that is called a quantum tunneling phenomena. So I try to assay that in the codes. This is super, but super crazy. To be like 100% honest, I had all this idea for, again, 15 years or 10 years or whatever, and I was not believing in this crazy stuff till I had these results, because it's crazy, but the simulations shows that if I perturb the system, again, the DNA of the Mycobacterium tuberculosis, with these incoming signals, if I do not, sorry, because I forget to put this in the slide. If I do not use the Doppler effect, the probabilities, sorry, por favor, ¿puede no hacer ruido? I am back again. Where I was, okay. So what was crazy, and I forget to put here, is that if I do not introduce this Doppler kind signal in the model, the probabilities that quantum tunneling occurs get not altered, do not change. Sorry again, guys. Sorry again. Por favor. So I am back again. Sorry. So I was trying to telling you that I forget to put in the slides, but if I do not introduce the Doppler-like effect, the tunneling probabilities do not change. And that is amazing, because that means that the system needs this Doppler encoding time to make that the probabilities of quantum tunneling change, which implies that you are introducing mutations in the sequence, where in non-coding regions, not in coding regions. So the main conclusions, I hope that you are still here. The main conclusion is that DNA regions behaves different between coding and non-coding if you try to understand DNA as a quantum system. And then that time-structured input, which is these Doppler-like frequencies are introduced in the system, are able to produce this shift in the microtiming, which is related with the probabilities that quantum tunneling occur, and that implies a biological consequence, which is to jump this proton barrier that quantum tunneling occurs, and to introduce mutations in the sequence. So that was all, guys. I hope you understand something of that. So main limitation of the paper. CMB, I mean, I make all the models around 34 gigahertz. The truth is that when you go through atmosphere with this kind of signal, you have a lot of attenuation, and if you are thinking multicellular organism, you have skin, and you can now go really deeper. But I already solved this because I am a biologist and I forget astrodynamics in the model. The truth is that now I am replacing this with sun radiation, because I already think this is a model. This is an in silico model. So you can use any kind of input signal perturbation. You can use thermal radiation, you can use magnetic fields, you can use microwave background stuff, which I use. I think that is really, this is working with solar radiation, but I need the Doppler effect. So when I start to analyze this, the distance from the Earth to the Sun was not enough to give me enough strong Doppler effect to have good results. But I forget that the Earth is spinning, that you have a lot of relative motions between Earth and Sun. Astrodynamics. Now it seems that it's working in that well. The qubit mapping strategy, this is a huge other limitation, because you cannot map an entire nucleotide as a qubit. That is already wrong by design. why this is still published and it is still because the results, because even with this huge limitation, the results are telling you that even with this, sorry, again, a huge limitation, DNA is behaving in a correct way when you compare with controls. The correct way to try to do that is map the protons of the different nucleotides directly to qubits. Why I not try that? Because I don't know. Again, this is my own, me by my own. So now I start to collaborating with other persons that are saving this. With the quantum computer guy from Aalborg. Then the simplified quantum dynamics. This work with the Schrödinger equation and Hamiltonian, and you can really get more complicated here. Again, I have limitations as a scientist. The tunneling model can be more realistic. You have stuff. What is the next stage? Try to solve that limitations. How? First, try to, all the simulations have a lot of parameters. You need to fix a lot of parameters. So you need to make variations and you need computational power to do that. So first is strain the computational power, which I am trying to collaborate with people. Then, if you do one, you will be able to generate stronger predictions for sure. And then try to test these ideas in the wet lab. I am already working on some codes that this is a really beautiful experiment to try this in the wet lab. If you use CRISPR to modify the genome of a bacteria, I am making some codes and some programs that are able to predict the mutational rates in some experiments that are called short evolution experiments, that you can do that in bacteria. But that will be amazing because that will be a wet lab proof of this that will have really a lot of implications in aging, for example, or cancer. Or these have really a lot of potential applications. So in the moment that the DAO start to fund other entities through the Aalborg University, for sure, I will share with you a proposal. We are asking for this amount for 2 years. And then finally, if you want to explore this idea beyond the paper, guys, I already write a science fiction saga of this, because this kind looks as science fiction. So if you look the Antenna of Dream in Amazon, you can buy this name one book. Is a short story about all these ideas, but in a science fiction story, and it's mixed the artificial intelligence. This signal coming from humans from the future, and I don't know. I imagine that people in the DAO will like this kind of crazy ideas, and then I create this, and we can really argue and discuss about this, because this is a platform that use the ideas on this in this paper to make transformation of DNA reports into music, fractal art, and entropic barcode. So this is the last slide. So if you go to shempita.org, you will see the web page, but imagine that you did some DNA analysis in these 23andMe sites or whatever. So that is related with a raw data report. You can ask that data report and you upload that in shempita.org, which is my website. And you can produce fractal image of your DNA directly from your report. You can produce music, or this that I think that is from interest for the DAO, which is entropic barcode. It's based on 268 military grade blockchain credential. It's blockchain ready. You can create a personal NFT from here directly from the website. you don't never upload your report to the website. I mean, we can argue, but if you guys are interested in some business ready model already working, based on quantum biology, based on crypto, and more important, based on science, super happy to collaborate. I am charging $199 now in this moment. So thank you for listening. I hope that you like it.